Vanakkam. Welcome to this course on classics in neuroscience. In this series of videos, we are looking at classic Nobel Prize winning works. In this series, starting with this video, we will look at the classic Nobel Prize winning works of Edgar Adrian, whose picture is given here. In this video, we will look at how Edgar Adrian essentially pioneered novel instruments to make measurements of the physiological or the neurophysiological system, and how he started collaborating with the brilliant Keith Lucas, and how this collaboration led to several fruitful adventures and several fruitful outcomes for Edgar Adrian. So, for Edgar Adrian, in 1932 he remarked that the history of electrophysiology has been decided by the history of electrical recording instruments. So, for Edgar Adrian, everything is about measurements, everything is about recording the electrical activity in the neural system. Early 20th century researchers, including Edgar Adrian, had several challenges. Like, you really cannot measure electrical activity with that much precision, accuracy and confidence. There were many challenges. Really much of the work itself and the outcomes depended entirely on the instrument that is used and the instrument sensitivity. So, Adrian and several others, we will see several other colleagues who contributed to Adrian's research and who also went on to win their own awards, for example, contributed to the development of devices to make measurements in this system. So, to a large extent, these recording systems determine the success or failure of the experiment. This still is true, but this is more true, or this was more true in the early 20th century, in the 1910s, 1920s, 1900s and so on. Two of the most significant instruments used in the late 19th century and the early 20th century were the capillary electrometer, which is primarily used in the late 19th century and the early 20th century, and the string electrometer, which was the instrument of choice later in the early 20th century. The pictures are given here. The capillary electrometer was developed by Gabriel Lippmann and Etienne Jules Marey in the 1870s. This was useful to record small changes in electrical activity, and this was further improved by Francis Gotch and Victor Horsley, who also worked on a similar topic and who came up with devices that can detect really small changes, electrical changes in nerves, and they found that some evidence for the presence of a refractory period, a period during which no action potential can take place. There are several drawbacks of the capillary electrometer. One primary drawback was that the capillary electrometer cannot measure fine features of the nervous system, and was not essentially, did not actually have that resolution and the accuracy. Of course, let us remember this was the late 19th century. Then the string galvanometer, which was pioneered by Einthoven in the early 20th century, was much better than the capillary electrometer, but the problem was that it still did not provide the sensitivity that was needed for the study of neuronal impulses that Edgar Adrian was interested in. To understand how Edgar Adrian and his studies proceeded and his instruments and how he got interested in instruments, we will have to study about his teacher and mentor Keith Lucas. Keith Lucas was born in 1879. he was completely fascinated by mechanical and electrical instruments from a very young age, and he attended Trinity College, Cambridge, where he was interested in physiology. So Lucas was interested in understanding muscle contractions. Specifically, he was interested in understanding why muscles can flex partially. That is, assuming that one muscle is a whole, if it flexes, then the whole muscle should flex. Why is it that some partial flexion is possible? So, in the good old days, it was not perhaps known that the possibility that only some muscle fibers could contract was not known at that time. So, Keith Lucas, through his experiments on frogs, showed that partial muscle contractions happen only when some muscle fibers contract fully, suggesting the all or nothing theory of muscle fiber behavior. That is, whenever the muscle fibers get the stimulus, they will contract, and when they don't, they won't contract. But a group of muscle fibers may contract, but other groups may not contract. So, that is all or none behavior of a group of muscle fibers, but not other fibers, because those other fibers are not stimulated. So, frog was the chosen specimen because it was easier to isolate muscles and nerves on frogs. For the longest time, this field of study of movements has used frogs as the usual specimen, because it is easy to isolate specific muscle. For example, the frog sartorius muscle is a famous specimen that is used to study biomechanics and the mechanical properties of muscles. It's a different topic, not in this field, not neuroscience, more mechanical aspects. For the longest time, this field of science also used to be called as the frog science, because a lot of people continue to study frogs. So, it is easier to isolate specific muscles, and also easy to isolate neuronal supply to these muscles. So, Keith Lucas continued his research to nerve physiology from muscle physiology. He asked the question, do neurons or nerve fibers also obey the all or nothing law? Is it that when neurons fire, they fire, otherwise they don't fire at all? Is that the case? Yes. So, in this process of studying nerve physiology or neurophysiology, Keith Lucas practically redesigned the capillary electrometer, thus increasing its sensitivity, and he combined it with an instrument that can deliver measurable electrical shock. So, you can stimulate and then you can measure its outcome. Very similar to the approach used by Charles Sherrington, who would stimulate and then measure the outcome with timing, with some good measure of timing between them. So, when you do that, you were in a position to study how stimulation could predict a specific response. So, you can come up with a one-to-one response, one-to-one relationship between stimulation and response. So, with his instruments that he developed or modified and improved, he was in a position to bring in a new assistant. He was looking for a new assistant in his lab. He was essentially looking for somebody who was bright, young, and who would be very happy to help with his experiments. And that's when Edgar Adrian entered the scene. So, Edgar Adrian joined Keith Lucas's laboratory as an assistant, where Lucas was immediately able to recognize Edgar Adrian's talent and enthusiasm, because Edgar Adrian was another brilliant, gifted and very interested student of physiology. So, Lucas gave the task of finding out whether the all or nothing principle that he observed in muscles or muscle fibers also applies to nerves or nerve cells or neurons. And Adrian came up with a novel method whereby he will use alcohol vapors to test nerve impulses. So that means there is a particular critical amount of alcohol to use, just enough amount of alcohol to weaken the impulse but not completely stop it. But once you remove the alcohol block, the impulse must jump back to the original value, suggesting that the all or nothing principle also applies to the neurons. So, brilliant approach that Edgar Adrian used along with Keith Lucas. With this, we come to the end of this video. In this video, we looked at how novel instrumentation or electrical recording devices essentially guided discovery of fine principles of neurophysiology as demonstrated by Edgar Adrian and Keith Lucas, and how Edgar Adrian came in contact with Keith Lucas and performed this brilliant experiment to suggest that the all or nothing principle also applies to nerve impulses and not just muscles or muscle fibers. With this, we end this video. Thank you very much for your attention.